and some are saying okay. Now I think it is clear. I am good, I am fine, I am very well. Okay students, so in last class we discussed about calorimetry. We discussed that if we supply heat to any body, then two possibilities are there except thermal expansion. Its temperature can change or its phase can change. To both we understood when phase will change and when temperature will change, both we understood. If temperature changes, then you have to use this formula delta Q equal to ms delta T. And if phase changes, then how much heat we supplied? Q equal to ml, right? Okay, so both we understood in last class. Today we will discuss ideal gas laws. Okay, so first, which gas is ideal gas? Okay, all gases are not ideal. For ideal gas, some conditions are there. If a gas follow those conditions, then that gas becomes ideal gas. Okay, so these are the laws. A gas must follow these laws, then that gas becomes ideal gas. Okay, so first law is Boyle's law. Okay, to understand this Boyle's law, and this is important for NEET exam also. Okay, so Boyle's law. First statement is written, you all can read this statement. According to it, for a given mass of an ideal gas at constant temperature, what is the condition? Constant temperature. What is the key point here? Constant temperature. So this is key point, right? So wherever such type of points are there, so these key point you have to underline, right? Constant temperature. Okay. So according to it, for a given mass of an ideal gas at constant temperature, the volume of gas is inversely proportional to pressure. You know very well what? You all use balloon, right? In balloon you filled air also. Okay. So a balloon you have. Now what you are doing? You are compressing the balloon. Tell me, can you compress the balloon? You will say yes. And when you compress the balloon, so its size decreases. Agree or not? Yes. If size decreases means volume decreases. Agree or not? So its volume decreases. Okay. And if volume decreases, so what about pressure? So at the same time, how this volume is decreasing? So you are compressing it, means you are applying external force. Agree or not? So you are applying external force, and pressure formula you all know, pressure equal to force divided by area. Means force produce pressure, yes. So we apply extra force, means we apply extra pressure on the balloon. If we apply extra pressure, so what will happen? In front of us we can see that balloon size decreases, means balloon volume decreases, right? So pressure we are increasing, to volume decreases. Agree or not? Okay, so we can say that pressure and volume both are inversely proportional, but it happen only if temperature is constant. So condition is what? Temperature must be constant. Then volume and pressure both are inversely proportional. Means if pressure increases, here we can understand, we can write it like this also, pressure is inversely proportional to volume. So if volume increases, then pressure will decrease, right? And reverse that, if volume decreases, then pressure increases. actually here one point that first we have to increase the pressure, then only volume will decrease, right or not? So we have to increase the pressure, then volume decreases. I given the example, balloon example. Okay. So if we draw graph between pressure and volume, so this graph is like this. How? Yes, we can say that if we move this side, the volume will increase. If we move this side, the volume will increase. If volume will increase, the pressure must decrease. So you can see this graph is moving down, and on the y-axis what is there? Volume. Otherwise you can check. You can take any volume. Suppose this volume. This volume is V1. How much pressure here? Pressure is on y-axis, so pressure is P1. Okay. Now take one more volume. This volume is V2. How much pressure? Pressure is P2. Now tell me which volume is higher? V2 is higher. Okay. Which pressure is higher? Which pressure is less? If volume V2 is higher, then corresponding to volume V2 pressure P2. P2 is less than P1. So we can say here that V2 is greater than V1. V2 is greater than, corresponding to it pressure is less. So this P2 is less than P1. Means both are inversely proportional. Understood? Is it clear to all of you? Okay. So we understood it. Now one more graph is there. In NEET they ask different graph. Pressure volume graph we understood. Sometimes they ask graph between PV and V. Graph between PV and V. So how to know? Understand. So what is the condition? Condition is that, understand this point. Condition is what? That volume is inversely proportional to pressure. Okay. So yesterday also we discussed that if we remove proportionality constant, if we remove proportional signs, then we have to remove it by using a constant. Agree or not? So here also if we remove this proportional sign, then it must be V equal to constant by P. What is C here? C is proportionality constant. Agree or not? Okay. Now if we draw graph between volume and pressure, so we understood this graph is like this. But here this graph is given between PV and V. Okay. So how to draw graph? Understand this point. This is very important. Understand everyone. Understand everyone how to draw graph. Okay. So first you have to check on y-axis what is given. On y-axis what is given? This is y-axis, you know. This is y-axis. Okay. And this is x-axis. Yes, you all know it. So on y-axis what is given? PV. And on x-axis what is given? V. Okay. So what you have to do here? Transfer P this side. Then it will become PV. Okay. So PV equal to constant. If we transfer this side to PV equal to constant, means product of pressure and volume is not changing. You agree or not? PV equal to constant means product of P and V, pressure and volume, is not changing. Whatever you do, it will remain constant at constant temperature. Agree or not? And for a given mass of an ideal gas, this one also you have to remember that mass of ideal gas must be constant. Both are the condition. Is it clear or not? Okay. So PV is constant. Whether you increase pressure or you decrease volume, you increase volume or you decrease pressure. Whatever you do, PV will not change. That's why graph between PV and volume is a straight line. Why straight line? Because PV is not changing. PV equal to constant. You agree or not? Here we can see this PV on y-axis. What is there? PV. So this is y. Okay. This is y. Y equal to constant. C is constant. Agree or not? So you can see here this y value is constant here. Okay. This y value is constant. This is constant. Here also, here we drawn graph between PV and V. Here we drawn graph between PV and P. Both are constant. PV is not changing by changing pressure. PV is not changing by changing volume. Why? Because PV is constant. So this is constant. Here also we can see this is what? Constant. Clear? Okay, this is y axis and this is x axis. Understood you all? So we understood here graph between pressure and volume first, then PV and volume, then PV and V. Now one question for you. Can you draw graph between pressure and 1 by V? Okay. What I am asking, graph between pressure and 1 by V. Okay, you can see here. What I am asking, graph between pressure and 1 by V, not V. Here V is there. Now I am asking pressure and 1 by V. Can you draw graph? You can send your response that this graph, sir, it will be hyperbola, parabola, like this. Very good. One girl, this Ratna. Yesterday also this girl given response, I remember. This girl's given correct answer. Very good, Ratna. And this, okay, Ratna, yes, very good, correct. And this Skotivara Koteswara, your answer also, Venkateswar, correct, very good. And this Madhu also correct, very good. You all are good students. I am very happy that you are given correct response. Very good. All are saying straight path, straight line. Very good, very good. Okay, so how to draw graph? Very simple. You have to just check on y-axis what is there, on x-axis what is there. Means here y-axis pressure is there, so this pressure becomes equal to y. This pressure becomes equal to y. Okay, because this pressure is on y-axis. 1 by V is on x-axis, so 1 by V becomes x. Agree or not? Okay. Now what is the condition? Condition is, we know the condition. Condition is V equal to C by P. This is what? Boyle's law. Okay. We know the condition. Now here put the values. So 1 by V is x. To do one thing, transfer P this side. So it is do one thing here, transfer this P this side, V this side, so it become P equal to C by V. Okay, now P is Y, and 1 by V is X. 1 by V, we can write it 1 by V into C. So 1 by V is X and C also. Okay. Otherwise Y equal to Cx. So this is what? This is straight line. Y and X both are directly proportional. So this graph is straight line. You agree or not? So graph will be straight line. Right? Okay. So we understood graph between pressure and 1 by V will be straight line. Okay, so we understood Boyle's law. What are the conditions? If mass of ideal gas is constant and temperature is constant, then we can say that pressure and volume both are inversely proportional, and we know very well it. If we compress the balloon, then balloon size decreases, means balloon volume decreases, and we are compressing it, means we are applying extra force, means we are producing extra pressure, because force produce pressure, pressure equal to force by area. Okay, so we understood it. Now next is Charles' law. What is the condition? What you have to mark here? What is key point here? So key point is, read the line. According to it, for a given mass of an ideal gas at constant pressure, so this time pressure is constant, volume of gas is directly proportional to absolute temperature. So this time what they given, that if constant pressure is there and given mass also fixed, right? We are doing experiment here for a particular amount of ideal gas. Okay, and if we maintain constant pressure, then we can say volume and temperature both are directly proportional, right? If we heat up the balloon, then what will happen? Say, you all seen hot balloon, hot gas balloon, you all seen, right? So if we heat up the balloon, then what happen? You will say, sir, that balloon expand, right or not? So if heat up means increase in temperature, right? So if there is a increase in temperature, then if it will expand means we can say that its volume will increase. So at constant pressure, and we are doing all these things in surrounding, in atmosphere. So in atmosphere, whatever is pressure is there, that pressure is same, constant it is. So pressure is not changing. So we can say that directly proportional to temperature. Volume is directly proportional to temperature. If we increase the temperature, then volume increases. So here graph, easily we can understand that graph will be straight line, right? Between volume and temperature. Between pressure and volume, it is inversely proportional, but here straight line. Okay. So graph between volume and temperature is a straight line. Understood? Now here V by T and V, and here V by T in T. So how to remember it, understand? So we know what? V is proportional to temperature. So again here also same. What we have to do? Remove the proportional sign. How? By using constant. So we can write it V equal to Ct. What is C here? C is constant. Agree or not? Okay. If we draw graph between volume and temperature, then both are directly proportional. If proportional, then graph will be straight line. Okay. If we draw graph between V by T and V, then it will be, to understand, divide this T this side. So it become V by T equal to C, and what is C? C is constant. So again same thing that we discuss here, that PV is constant. Here V by T is constant, means whatever we do, we change temperature or volume, but their ratio V by T will not change, it will remain constant. So V by T is constant. This is what? V by T, and it is constant, it is not changing. Agree or not? Here also this V by T is constant, it is not changing. Is it clear? So whether we change the volume or temperature, but V by T will not change. Why? Because it is the ratio. If we double the temperature, then volume also doubled, then ratio will remain constant. If we half the temperature, then then volume also become half. If volume become half means again ratio will remain constant. Is it clear to all of you? Okay, so we understood this Charles law also. Now next is Gay-Lussac law. What is it? Read the statement. According to it, for a given mass of an ideal gas at constant volume, what is the condition? Constant volume. You can see first constant temperature, then constant pressure, then constant volume. You know, for ideal gas, for any gas, we define three quantities. These are required, necessary to understand the gas behavior. What these are? Pressure, volume, temperature. Okay, so first constant temperature in Boyle's law, then in Charles' law constant pressure, then in Gay-Lussac law constant volume. And in all three laws, given amount of ideal gas means whatever is the amount mass of ideal gas, that is constant throughout the experiment, it is not changing. So again condition we know, given mass, okay, and constant volume. So here we maintain the temperature constant, then remaining quantity pressure, volume, constant pressure, then remaining quantity volume and temperature, constant volume, then remaining quantity pressure and temperature. So if volume is constant, then we can say pressure is directly proportional to absolute temperature, P proportional to T. Now this Charles law graph and Gay-Lussac law graph, only one change. What? T as it is, at the place of volume pressure is there, at the place of volume pressure is there, at the place of V by T it will be P by T, V by T it will be P by T, other are as it is. To graph also as it is, similar graph. Agree or not? Okay, so here this is y-axis, and at y-axis what we are taking? Pressure and temperature both are directly proportional, to graph will be straight line. Again they are taking here P by T, so same thing we have to do again. P is proportional to T, we have to remove this proportional sign, then P equal to CT. Okay, what is C? Constant. What is C here? C is constant. Okay, so P and T directly proportional, so graph is straight line. Now next P by T, so we need P by T. To do one thing, divide T this side. So P by T is equal to C, and C is what? Constant. Means again ratio of pressure and temperature remain constant. Whatever you do, always it is constant if we maintain constant volume and given mass. Agree or not? So if we change pressure, then also P by T will not change. If we change temperature, then also P by T will not change. But if we change volume, then it will change, because P by T is constant if volume is constant. If volume is not constant, then this condition is not possible. Is it clear? Understood? So you all understood it? Okay, just you can see these all three laws, because these are very important, and further we will solve questions. Okay, completing one more law we have to complete, then we will solve questions. Okay, next is Dalton's law. To understand it, Dalton's law, partial pressure. So what is it? According to it, the pressure exerted by the mixture of non-reactive gases is equal to the sum of partial pressure of each component gases present in the mixture. Suppose a mixture is there, in that 2-3 gases are there. So if we talk about, suppose in this room is closed, okay, and in this room we filled 2 gases, one is oxygen and another is nitrogen, okay. So oxygen gas will apply pressure here, yes, each gas apply pressure, okay. Nitrogen gas also apply pressure, yes. Then total pressure will be pressure due to nitrogen gas plus pressure due to oxygen gas. So this is Dalton's law. It is not a rocket science, very simple it is. What? That in the mixture, total pressure is equal to the sum of partial pressure of each component of gas. Partial pressure means pressure due to individual gas. Pressure due to nitrogen plus pressure due to oxygen equal to total pressure. Agree or not? One example, this as example you can understand. Suppose in this container 2 gases are there, O2 and N2. Okay, so total pressure is equal to pressure due to oxygen gas plus pressure due to nitrogen gas. This is known as partial pressure also. Partial pressure means pressure due to any one gas. That is the meaning of partial pressure. Okay, partial pressure means pressure due to individual gas. Clear? Okay, so we understood Dalton's law also. Now we will solve question. So you all understood these all laws, I believe, and now we will solve questions. So let's see how many of you will give correct response. Okay. Shall we start question solving? Okay, students. Answer also given, but be a genuine student, honest student, okay. First question. What is given? The pressure of a given mass of a gas. What? First, how to solve questions? To understand this point. Given mass, this is the key point. So you have to underline given mass, because in all these laws we discuss given mass, okay, of a gas filled in a vessel of volume V. It is filled in a vessel of volume V, okay, at constant temperature. Okay, temperature is constant, so initial volume V and temperature constant. If temperature is constant, then what will come in your mind? Which law will strike in your mind? Boyle's law or not? Agree or not? So just by reading it in our mind, what will strike? That it is related to Boyle's law. Agree or not? If it is related to Boyle's law, then we have to use Boyle's law condition. And what is that? Pressure and volume both are inversely proportional, right or not? Otherwise P equal to C by V. This is going in our mind, we are not doing anything. This is just we are thinking after reading this point, right or not? Okay, is reduced to 1 by third of initial volume. So what is given? Initial volume, that is how much V, then final volume, that is how much V by 3. Agree or not? These are given quantities. Calculate the percentage change in volume. No. Pressure is reduced to 1 by third. They given that pressure is reduced to 1 by third. Okay. So actually here pressure is reduced to 1 by third. So V final we don't know. Okay. We know, suppose P initial is P, then P final is given P by 3. So V final we can calculate by using it. Agree or not? Because PV multiple is constant. If it is constant, then we can say what? PV, product of pressure and volume, is constant, means initial equal to final. Constant means quantity is not changing. Quantity is not changing means whatever it was earlier, that is now. Okay, means P initial V initial is equal to P final V final. Right or not? This condition. So here pressure is given, okay, P final is given P by 3, to put the value. So P initial P, V initial V, P final P by 3, and V final we don't know. So P cancel out, 3 transfer this side, so V final value is how much? 3V. Agree or not? So volume becomes 3 times. Is it clear? Okay. Now students, this is the percentage change. You will see in physics, in many chapters at multiple places, you will see this type of question: find percentage change, find percentage change. Okay, so how to find percentage change? Understand. Percentage change formula is, this formula you have to remember. Percentage change formula is final quantity minus initial quantity divided by initial quantity. This is percentage change, and percentage change we are solving, so multiply with 100. Okay, so final volume minus initial volume by initial volume into 100. This is the formula of percentage change. Is it clear? Okay, so here we need final volume, already we calculated, that is 3V. Okay, initial volume, that is V, initial volume V multiplied by 100. So you know 3V minus V, 2V. Okay, so it become it become 2V by V into 100. V cancel out, so answer is 200 percentage. Is it clear? This is answer. Understood everyone? Okay, now this question. This question we will solve later, because we till now we did not discuss ideal gas equation. Okay, so we will solve this question later. Okay, it is clear. Now next, this question is little bit tough. Okay, so I will share these questions as a homework in our Telegram group. So you have to try this question and this last question also, because here they are using ideal gas equation. So first we have to know that ideal gas equation how to form. To understand this point, ideal gas equation, very important. We discuss three laws. By using those laws, because those are for ideal gas, by using those only we will form ideal gas equation. Okay, what is it? Understand. And here also this point you have to remember that pressure is proportional to number of moles. Pressure is proportional to number of moles. Okay, so we discussed there that pressure is inversely proportional to volume. Where? Boyle's law. Okay, equation number 1. Then we discuss pressure is directly proportional to temperature. Which law? We discuss it, you know, pressure is directly proportional to temperature, Gay-Lussac law, right? Okay, equation number 2. Okay, and pressure of any gas is directly proportional to number of moles. Pressure of any gas is directly proportional to number of moles. Here N is number of moles. If we combine these three, then it becomes pressure is proportional to T and pressure is proportional to N and T inversely proportional to volume. If we remove the proportional sign, then here also we need a constant. That constant is R. Here we will take C also, but we given this constant name R. So it is P equal to N R T by V. Otherwise we can transfer V this side, then it become PV equal to nRT. This is the ideal gas equation. This is the ideal gas equation. This one you have to remember. What is R here? R is constant and this R values also fixed because it is constant. So these values you have to remember. Is it clear? So this equation is ideal gas equation, PV equal to nRT. This N is number of mole here, R gas constant and P pressure we already discussed, V volume and T temperature. By using all these law, finally we form this equation. Is it clear? So in these two questions, question number, example number 2 and in this example, this ideal gas equation we have to use. So you have to try these questions at your home because today time completed. Okay friends, so I hope you all enjoyed the class and you understood today gas laws and ideal gas equation. Okay, thank you so much for listening the class. Further we'll discuss in next class.
Good morning, students. So in last class what we have discussed, electronic concept of oxidation reduction and basics of oxidation number, how to calculate oxidation number. Now yesterday you have got that test of mole concept. How many students have attempted those questions? Only those students have to tell that how many students have attempted those 20 questions. I attempted. Okay, very many students have attempted, na? Okay, very good. But you don't have answer key, na? So I will give you that answer key. Write down fast. Questions will not discuss. Only I will give you answer key right now. So when you come here, after reading complete chapter in detail, you will get all those questions. But this is only for your practice. So I will write answers for all those questions. For question number 1, option B. Second is D. Third B, fourth A. Fifth is B, sixth A. Eighth B. Ninth C. 10th B, 11 question C. 12 B. 13 A. 14 B. 15 C. 17 B. 18 D. 19 D and 20 B. So this is answer key. Now if you are able to attempt 50% of the questions, because in mole concept we have not discussed complete chapter here, we have done only basics here of stoichiometry, concentration terms, not very typical question. But still, if you are able to attempt 50% of these questions, it means you have listened classes very carefully. That is good if you are able to score 50% marks in this. Now you should keep these papers with you when you come for offline classes, and when you read that complete chapter, then you will see you can able to solve all these questions. Okay. Now let us start today's class. So we'll start about rules for calculation of oxidation number. We have written 2 rules. So we'll write in brief those 2 rules, then move to third. So what we are discussing, rules for calculation of oxidation number. Okay. So in last class we have discussed 2 rules. First we have written, if any element in its free state or elemental state, then what is oxidation number assigned to them? Oxidation number is equal to 0. Clear? And second rule we have written that oxidation number of any cation or anion is equal to charge present on it. And this is not valid for polyatomic ions. Not for polyatomic ions. So if you remember, in last class we have written these 2 rules. First rule we have written for free state or elemental state, that oxidation number is 0. And next rule we have written oxidation number of any simple cation or anion is equal to charge present on it. So if you write examples, for example, F negative, what is oxidation number? -1. This is simple anion. Sulfur 2 negative, what is oxidation number? -2. If I write magnesium +2, what is oxidation number? +2. So these are simple anions and oxidation number is equal to charge present on it. Now let us move to third rule. So these two we have discussed in last class. Now third rule is, if we take alkali metals in periodic table, if we take lithium, sodium, potassium, these elements are called as alkali metals. So I am writing here which elements: lithium, sodium, potassium, rubidium, cesium. In its combined state, remember what we are writing, combined state, not elemental state, shows which oxidation number? +1 oxidation number. Clear? So whenever they are forming compounds, alkali metal, they shows what oxidation number? +1 oxidation number. Let us take some examples. First example, I am writing compound for sodium. Let us take sodium chloride. Now in sodium chloride, what is oxidation number of sodium? +1. Then oxidation number of chlorine will be -1. So alkali metal in its combined state is showing what oxidation number? +1. Let us take any other example. If I take K2CO3, what is name of this compound? Name of this compound is potassium carbonate. Now if I break this compound into ions, how to break this? What is present here? K positive. And which ion is present here? Carbonate ion. We have no idea about any carbonate, carbon oxygen, leave that. What we need only? Potassium. So here what is oxidation number of potassium? It is +1. So whenever alkali metal forms compound, means it is in combined state, they show what oxidation number? Always +1. Clear? Now if we move to next rule. Next is for alkaline earth metals. Alkaline earth metals. Now group 2 elements are called as alkaline earth metal. Here we write beryllium also, but actually beryllium is not considered as alkaline earth metal, but we are writing for group 2 elements, we consider here beryllium also. So beryllium, magnesium, calcium, strontium, barium. These two are group 2 elements. So whenever these alkaline earth metal will form compounds, they form which oxidation state? +2 oxidation state. In its combined state, shows +2 oxidation state. So let us take example for these compounds. Let us take compounds, very simple, magnesium chloride, MgCl2. So if I break this magnesium chloride into cation and anion, this is ionic compound. Magnesium +2, and what is present here? Cl negative. So what we can say here, oxidation number of magnesium in MgCl2 is, what is oxidation number? +2. Because it is alkaline earth metal, so it shows which oxidation state? +2. Let us take one more example. CaSO4. So what is name of compound? Calcium sulfate. Now if we break this, so what is present here? Calcium +2 and SO4 2 negative. So oxidation number of calcium in this compound is, what is oxidation number? +2. So alkali metal has 1 electron in outermost shell, so they lose 1 electron, and what is formed? Cation having +1 charge. Alkaline earth metal have how many electrons in outermost shell? 2 electrons. So they will lose 2 electron, and what charge is present? +2 is present in its combined state. So these 2 points we have to remember. For alkali metal, oxidation state is +1. For alkaline earth metal, oxidation state is +2. Now next move to hydrogen. So generally hydrogen shows +1 oxidation state in its compound, except which one? except ionic hydrides. Now, what is meaning of ionic hydride? Now, this is important. So, meaning of ionic hydride is whenever hydrogen combine with any S block element. S block element means alkali metal and alkaline earth metal. So, whenever any S block element combines with a hydrogen, that is called as which hydride? Ionic hydride. And in ionic hydride, what is oxidation number of hydrogen in these compounds? It is -1. Generally, it will show +1. But in case of ionic hydride, that is S block element plus hydrogen compound, what is oxidation number of hydrogen? It is -1. So, if I take example, first example, let us take HCl. Now, HCl we all know it is H positive and Cl negative. So, what is oxidation number of hydrogen here? Oxidation number is +1. So, hydrogen is +1, chlorine is -1. Now, this is simple. Now, in case of ionic hydride, let us take example of any ionic hydride. NaH. Now, sodium is which metal? Alkali metal, and it is S block element. S block element plus hydrogen, it is ionic hydride. So, in case of ionic hydride, when you break, alkali metal always shows +1. Then hydrogen has to show what oxidation number? -1. So, in this compound, what is oxidation number of hydrogen? It is -1. Clear? So, this we have to remember that in case of hydrogen, generally it shows +1. Generally it shows which oxidation number? +1. But in case of ionic hydride, S block element plus hydrogen, oxidation number of hydrogen is how much? -1. Clear? Any doubt in this hydrogen? Now, let us move to next point, sixth point, that is for halogens. So, generally halogens, which are halogens? Fluorine, chlorine, bromine, iodine. In its compounds shows -1 oxidation state. So, what is general oxidation state? What is neutral compound? We'll discuss neutral compound. Wait for some time. Now, whenever its compound is formed, why that exception for hydrogen? It is not exception. Alkali metals and alkaline earth metals are electropositive than hydrogen. So, electropositive will show positive oxidation state, and electronegative, hydrogen is more electronegative than that. So, alkali shows positive, hydrogen will show then negative. Remember this thing, more electronegative element will get negative charge and electropositive will get positive charge. Okay. Now, generally halogen shows which oxidation state? -1. Now, in complete periodic table, most electronegative element is fluorine. So, what we write? Fluorine in its compounds always shows -1 oxidation state. Fluorine cannot show any other, only what we have to write? -1. But in case of chlorine, bromine and iodine, they can show range -1, 2, +7. So, they can show range. If it is more electronegative, then -1. If they are less electronegative, then they can show positive oxidation state also. Here, what is neutral compound? And compound does not have any charge, that is called as neutral compound. What is difficulty in this? CO2. Sir, then acetylene, that is radioactive element. You do not find any compounds of acetylene. You will find compounds of only these: fluorine, chlorine, bromine and iodine. So, for radioactive elements, we are not discussing here. So, chlorine, bromine, iodine can show a range of oxidation state, that is -1, 2, +7. So, if we take examples, Let us take NaF, sodium fluoride. So what is oxidation state of sodium here? +1. Oxidation state of fluorine is -1. Similar example, if we take KCl, so potassium will show +1, chlorine will show -1. So this is general. But if we take more electronegative element, this is very good, ClF3. Now you have to tell what is oxidation number of chlorine and what is oxidation number of fluorine in this compound. ClF3. In ClF3, you have to tell oxidation number of chlorine and fluorine. Ratna, chlorine +3, very good. Because remember here, fluorine is more electronegative than chlorine. So more electronegative will show which charge? -1. Now 3 fluorine are present, so total charge -3. So here chlorine is present in which oxidation state? +3. And fluorine is present in -1. Remember there are 3 fluorine. So 1 fluorine -1, total 3 fluorine -1. So chlorine is present in +3. So we can say here that chlorine, bromine, iodine can show range of oxidation state -1 to +7. And this fluorine always shows which oxidation state? -1, because fluorine is most electronegative element in the periodic table. Okay. Now if we move to next element, that is oxygen. So what we write generally, oxygen shows -2 oxidation number in its compounds. Okay. So generally oxygen shows which oxidation number? -2, and oxygen -2 is called as oxide. But there are some other compounds of oxygen which are formed, that is peroxide, superoxide, and when oxygen combines with fluorine, so different oxidation number are possible. Remember, for O2, one simple theory is used, that is molecular orbital theory, that we will study in chemical bonding. So O2 will combine used as a molecule. We do not write oxygen atom, but we write O2 molecule for peroxide and superoxide. So if I write oxygen 2 negative, this is called as which ion? Oxide ion. And what is oxidation number here? -2. Clear? And next, if we write O2 2 negative, now remember O2 is a complete molecule having which charge? -2. We do not write that oxygen because O2 is a homonuclear diatomic molecule. For this bonding, what is used? Molecular orbital theory, that we will study in later part of chemistry, chemical bonding. So name of this ion is peroxide ion. So in peroxide ion, oxidation number is -1. It is oxidation number. 2 oxygen having -2. So for 1 oxygen, what is charge? -1. And next is superoxide, O2 -1. So when O2 gains 1 electron, which ion is formed? Superoxide. And what is oxidation number? -1/2. I am not writing oxidation state here, I am writing oxidation number. So oxidation number of oxygen in O2 negative is how much? -1/2. So remember, O2 2 negative is peroxide, O2 negative is called as superoxide. So if I write examples for these, let us take compound Na2O2 and one compound is KO2. You have to tell name of these two compounds. What is name of Na2O2 and what is name of KO2? Tell me name of these two compounds. Then we discuss about their oxidation number. Only names you have to tell, not any oxidation number. Name of Na2O2 and name of KO2. Na2O2 is sodium peroxide. Very good. What is name of KO2? KO2 is potassium superoxide. Very good. Now, how to identify it is peroxide or superoxide? It is very easy. Break these into ions. Now, how many sodium are present? 2. And what is charge on sodium? +1. So, total charge is +2. So, this O2 will get what charge? Total -2. And what is O2 -2? It is which ion? Peroxide ion. So, what is name for this compound? Can we say this is sodium peroxide? And in peroxide, what is oxidation number of oxygen? -1. Same if we break this KO2. How to break this KO2? Potassium shows +1. Only one potassium. So, what will be charge on 2 oxygen? It will be -1 because total charge will be same, positive and negative, because it is a neutral. So, K positive, O2 negative. Now, in O2 negative, what is name for O2 negative? Superoxide. So, oxidation number is -1/2. And what is name of compound? It is potassium superoxide. Clear? So, remember this. This peroxide and superoxide either you will find in acids of sulfur, oxo acid, and somewhere you find in this S block elements, potassium superoxide, sodium peroxide. And next important point is whenever oxygen combines with fluorine. Now, fluorine is more electronegative than oxygen. Fluorine is more electronegative than oxygen. So, fluorine shows which oxidation number? -1. Then oxygen has to show which oxidation state? Positive. So, if I write oxidation state in OF2, 2 fluorine -2. So, oxygen will show +2. Now, here 2 fluorine -2, not 2 oxygen. So, 1 oxygen will show +1. Now, in OF2, what is oxidation number of oxygen? +2. In O2F2, what is oxidation number of oxygen? +1. So, only there are 2 compounds of oxygen, OF2 and O2F2, where oxygen can show +1 oxidation state and +2 oxidation state. Reason is fluorine is more electronegative than oxygen. So, fluorine will show negative, oxygen will show positive. There is one more ion, ozonide ion, O3 negative, but that is not required. Ozonide, what is oxidation number? -1/3. If you want to write, I will write here, O3 negative. This is called as which ion? Ozonide ion. But you will find very less this ion, O3 negative. So, if I write oxidation number of oxygen, here it is -1/3. For 3 oxygen, -1. For 1 oxygen, it is -1/3. But this ozonide ion you will find very less. More important, what you find is oxide, peroxide, superoxide, and these 2 compounds of with fluorine, OF2 and O2F2. Clear? So, this is for which element? Oxygen. So, general oxidation state is how much? -2. Which is least electronegative. Alkali metals on moving down the group. So, if we do not consider radioactive element, then cesium is least electronegative. Metals are electropositive. Non-metals are electronegative. Just remember this term. And only remember 3 elements: fluorine, oxygen, nitrogen. Fluorine is most, then oxygen, then nitrogen. So, when this periodic table you will study, then you will find all these order for electronegativity, order for electropositivity. Now, let us move to next point. So, we have written all the elements whose oxidation number is fixed. Now, if we take any neutral molecule, let us take NaCl. Now, NaCl, there is no charge present on this compound. So, how many elements are present here? 2 elements. One is sodium and one is chlorine. So, if we take oxidation number of sodium, how much it is? +1. And oxidation number of chlorine is -1. So, what is sum of these oxidation number? It is equal to 0. So, whenever there is a neutral molecule or neutral compound, means compound does not have any charge, then sum of oxidation number of all atoms in that molecule is equal to 0. Now, this rule will be helpful in finding the oxidation number of any one atom whose oxidation number is variable. For example, if 3 elements are present in a compound, for 2 oxidation number is fixed, that we know, and for one element it is unknown, then sum of oxidation number is equal to 0. So what rule we write here? That sum of oxidation number of all atoms in a neutral molecule is equal to 0. Here. So first I am writing very simple example, NaCl. So if I write oxidation number of sodium here +1, and what is oxidation number of chlorine here? -1. So if I write sum of oxidation number, it comes out to be 0. Oxidation number of sodium plus oxidation number of chlorine, it is equal to 0. Now where this rule is applied? Now this rule will be applied for calculation of oxidation number of any one atom in a neutral molecule. And out of these three, for hydrogen oxidation number we know, for oxygen we know, what is unknown here? Sulfur. So we have to find this oxidation number of sulfur. Now how to write this? How many hydrogen are present? 2. And what is oxidation number of hydrogen? +1. Now next is one sulfur is present. For sulfur we do not know. Let us take X. Now after this, how many oxygen are present? 4. And oxygen shows generally which oxidation state? -2. Now sum of oxidation number is equal to 0. Listen again, what we have to take? How many hydrogen are there? 2. So 2 into, for one hydrogen charge is +1. Next sulfur is unknown. So what we are taking here? X. Now plus 4 oxygen, and oxidation number of oxygen is generally -2. So 4 into -2 is equal to 0. So when you solve this, what you get? 2 plus X minus 8 is equal to 0. So what is value of X? +6. So can we say oxidation number of sulfur in this compound is +6? Just like any doubt in this example, H2SO4. We have used this formula, sum of oxidation number of all atoms in a neutral molecule is equal to 0. Only this you have to check in H2SO4, any doubt? So if there is no doubt, I will write one example. HNO3. You have to find out what is oxidation number of nitrogen in HNO3. Oxidation state we will discuss in next class. Today we are discussing oxidation number. So focus on oxidation number. Tell me what is oxidation number of nitrogen in HNO3. Students are telling +5. Very good. And whenever you are writing oxidation number, do not say only 5. You have to tell +5 or -5. So answer is +5. So how many hydrogen? 1. For hydrogen, +1. For nitrogen, what we take? X. 3 into -2 is equal to 0. Clear? So what is oxidation number if you find? What we get? +5. So oxidation number of nitrogen in this compound is +5. Clear? Let us do one more example. H2S2O3. So you have to tell oxidation number of sulfur. H2S2O3. Shani, 5 is not possible. 5 is incorrect. 4 is also incorrect. Ratna, it is correct. What is oxidation number? What you get here? +2. Now here 2 sulfur are present. So what we take here? 2 hydrogen, 2 into +1. Now how many sulfur are present here? 2 sulfur. So on 1 sulfur, if charge is X, for 2 sulfur it is 2 times of X. 3 into -2 is equal to 0. So what you get here? 2x is equal to +4. So what is value of X? What we get here? +2. So what is oxidation number of sulfur in this compound? It is +2. Okay. So what we have to find, remember in this case that if 2 sulfur are given, we have to write 2 times of X. So X will come out how much it is? +2. So this is for which molecule? Neutral molecule. Now let us take one example or next point for charged species, that is polyatomic ion. So what we write, sum of oxidation number of all atoms in a charged species is equal to charge present on it. So if there is a polyatomic ion, then sum of oxidation number of all atoms is equal to charge on that polyatomic ion. So if I take example sulfate ion, SO4 2 negative. Now in sulfate ion, how many atoms are present? One sulfur is present, and how many oxygen are present? 4. And what is total charge on this polyatomic ion? It is -2. So sum of oxidation number of all these is equal to -2. Now if we do not know about sulfur, what we can take sulfur as X. How many oxygen are present? 4 into -2. So sum of oxidation number is equal to charge present. What is charge here? -2. So what oxidation number we get? +6. So oxidation number of sulfur in sulfate ion is +6. Now tell me answer for PO4 3 negative. In phosphate, oxidation number of phosphorus. Oxidation number of phosphorus in phosphate. I will write one more. And one we have to take Cr2O7 2 negative dichromate in chromium. These two we have to solve. Oxidation number of phosphorus and oxidation number of chromium. For phosphorus +5, correct. And solve for dichromate. Cr2O7 2 negative, name is dichromate ion. This PO4 3 negative is called as which ion? Phosphate. And this Cr2O7 2 negative is called as which ion? Dichromate ion. For dichromate, no, +12 is not correct. +6. +5 and +6. So how to solve? For phosphorus, what we take? X. How many oxygen are present? 4. So 4 into -2 is equal to -3. So it is -8. So what you get? +5. So oxidation number of phosphorus in phosphate ion is how much? +5. Now if we take dichromate, how many are present? Chromium 2. So what we have to take? 2 into X +7 into -2 is equal to -2. So what we get here? 2x -14, it will become 12 plus 14 minus 2. So what is value of X? +6. So in dichromate ion, what is oxidation number of chromium? +6. Why can't we take valency as charge? Valency is not for any molecule. Valency is defined for a particular element. Now this charge is present on complete Cr2O7, not on this chromium. And valency is for elements. It is not for any compound. Okay. So total how many rules we have discussed for calculation for oxidation number? Total 9 rules we have discussed. So today you have to revise all these rules for these 9. In next class, we'll discuss a lot of questions on this oxidation number. So if you remember all these rules, then only we can solve these. So today we are going to discuss only these rules, but in next class what we discuss, question based on this. Okay. Can we take valency for single element as charge? So if it is given, for example, if O2 negative, so now it is ion, so in O2 negative, oxygen -2, so that is its valency as well as charge. But valency is not negative, valency is only 2. So do not mix valency charge, just focus on this oxidation number. So today you will get a test for nomenclature. Tell for CrO5, Venkat, where you will go very fast. Now be very slow. So first focus on easy topics. I will give you a lot of examples which you have not seen anywhere else also. So today we'll discuss only up to this, revise these all topics. In next class we'll meet and practice more questions on this.
Hello, good morning students. Good morning all of you. Good morning. Good morning mom. Yes, good morning Afifa. Good morning Tanu. Yes, Joshita. Yes. Yes. Good morning. Good morning all of you. Yes, Anu. Yeah, good morning. Balu, good morning. Yeah, good morning all of you. How are you all? Stay hydrated. Be indoors. Are you following that? Yes. I'm good. Madhumita, good. I'm good. Yes, Kavya ready. Yes, I'm good. Yes. Okay, yesterday I forgot what I have taught you. Can you please tell me which topic you have learnt yesterday? Yesterday. Which topic? Yeah, good morning. Good morning. Nice sun, bright. Good. Monera. Oh, okay, good. Monera. Yes. Monera. Correct. So, Monera, does it include eukaryotes or prokaryotes? Monera, will it include prokaryotes or eukaryotes? Yeah, hi Sanjana. Yes. FIFA prokaryotes. Yes, Sanjana. Yeah, prokaryotes. Very good. Okay, prokaryotes. Correct. Can you give me examples of prokaryotes which you have learnt yesterday? Examples. Algae, fungi, bacteria. Which ones are prokaryotes among these three? Algae, fungi, bacteria. Yes, yeah. Dhiraja, yes, bacteria, correct. Himaja, yes, bacteria. Balu, eubacteria, yes, eubacteria. Yes, good. Bacteria. Right. Name the Moneran which is involved in nitrogen fixation, but it performs photosynthesis like plants. Tikshana. Yes. Yeah, Sindhu. Yes, I am good. Cyanobacteria. Yes. See all of you. Blue-green algae are nitrogen fixing photosynthetic photoautotrophic bacteria. So, cyanobacteria or blue-green algae, yeah, they are the ones which are going to do the function of nitrogen fixation. Very good, Sanjana. Heterocyst. Correct. They will do that in the heterocyst. Yes, correct. Blue-green algae. What is the other name of blue-green algae? Yes, Mallikarjuna. Good morning. What is the other name of blue-green algae? Cyanobacteria. These are only called as cyanobacteria. Okay, the last one before proceeding to today's topic. Name the wall-less prokaryote. Wall-less Moneran. Cell wall-less Moneran. Yes, cyanobacteria, correct. Yes, Anju. Yes, cyanobacteria, correct. Wall-less Moneran. Yes, very good, Koteshwarao. Yes. Mycoplasma. Yes. Okay. Can they survive without oxygen? Can they survive, that mycoplasma? Can they survive without oxygen? Or they can't survive? Can they or they are not? No, Chakri. They can't survive without oxygen? They can survive. Yes, they are anaerobes. Yes. Yeah, they can survive without oxygen. Correct. Among bacteria, cyanobacteria, mycoplasma, which are having simple structure but complex behavior. Options are bacteria, cyanobacteria, mycoplasma, which are having simple structure but complex behavior. Yes, Madhumita, very good. Sanjana, very good. What CO2 is doing, Taruna? Okay, eubacteria, very good. Eubacteria, true. Last question. With this, I'll go for the next. Okay. Name the moneron which can produce methane gas. Name the moneron which can produce methane gas. Bacteria, that means eubacteria, archaebacteria, cyanobacteria, mycoplasma. Which ones? Okay, Vasant, very good. Methanogens. They belong to which group? Madhumita, very good. Archaebacteria. Yes, correct. Methanogens are the archaebacteria which are going to produce the methane gas. Correct. Archaebacteria. Very good, very good. So, all of you are, though you are chatting, you are learning also subject. Very good, very good. Keep it up. So, as we have completed the first kingdom, that is Monera, I would like to proceed to the second kingdom based on the evolution order. You know, primitive, oldest ones are Monerans. Second group of organisms belong to the kingdom Protista. So, Protistan ancestors are Monerans. So, Protista totally, it is formed from the Monerans. Yeah, and the ruminants also, Sanjana, ruminants, methanogens are present. Very good. So, next group of organisms belong to the second kingdom, that is Protista. So, let us see in the Protista which organisms are present. Protista include all unicellular eukaryotes. It includes all unicellular eukaryotes. Okay. Because the organisms have been separated from the plant kingdom, animal kingdom, and from the Monera kingdom, from the plant kingdom, all those organisms have been separated, and Ernest Haeckel created Protista as a kingdom. Third kingdom is the Protista, which was created by Ernest Haeckel. So, he got some of the things like Chlorella, Chlamydomonas from the plant kingdom, and he got the amoeba, paramecium, and euglena from the animal kingdom, protozoans he took, and he prepared all these organisms by keeping them together. He prepared one more kingdom that is called as Protista. So, he created the kingdom Protista by taking some of the organisms from the plant kingdom and some of the organisms from the animal kingdom. But he took which organisms? All should be unicellular. They are all having only one cell and they are all eukaryotes. Okay. So, all unicellular eukaryotes have been separated from the plant kingdom and animal kingdom for the creation of Protista. Okay. So, this Protista include all the eukaryotic features. Eukaryotic features like if they have cilia or flagella, they will have 9+2 arrangement, cytoplasmic streaming will be there, histone proteins are present, all eukaryotic, well-defined nucleus is there, nuclear membrane is there, nucleolus is present, all the eukaryotic features are present in these Protistans. But scientists are having so much problems with this Protista because this kingdom doesn't have any perfect boundaries. They did not limit by any boundary. So, there are no well-defined boundaries are not there for this Protista kingdom. So, whenever we want to bring some other organism from any other kingdom, we can place it by following some characteristics. So, that's why all the people, based on the classification criteria, they used to take out one organism from one kingdom and they used to keep it in this. So, for this kingdom, there are no well-defined boundaries. No well-defined boundaries. Okay. Because this also shows the mode of nutrition, different types are there. Diverse mode of nutrition is shown by Protistans also, like Monerans. Okay. So, there are no well-defined boundaries for this kingdom. Now, this Protista kingdom has been divided into three groups. Protista kingdom, it is divided into three groups. photosynthetic protists. So the photosynthetic protists, they will be performing the photosynthesis. They are meant for photosynthesis. Okay. Then they have divided them into consumer decomposer protists. Then protozoan protists. Based on the mode of nutrition, okay, the Protista kingdom has been divided into 3 groups of organisms. First group, all of them will perform photosynthesis. So photosynthetic protists are kept under this. These are called consumer decomposer protists. Here the heterotrophic mode of nutrition will be there. Okay. Then protozoan protists, they'll show only the consuming nature of the protistans. So based on the nutrition, they have classified like this. Under each of these category, we have further types. In the photosynthetic protists, we have 3 types: chrysophytes. Chrysophytes examples are diatoms, okay, golden algae. They'll come under chrysophytes. Second category, okay, I can write here, dinoflagellates. Third category, euglenoids. So there are 3 categories under the photosynthetic protists. The first ones are chrysophytes. Under this, the examples of diatoms and golden algae will be there. And second group are dinoflagellates, and third group are euglenoids. Then in the consumer decomposer protists, we have, in this only one things are there, that is slime molds. In this one, the protozoan protists, we have amoeboid protozoans, amoeboids, flagellates, ciliates, sporozoans. This is the classification of Protista. So first, Protista will be classified like this. I hope all of you can see now. Protista kingdom, it is subdivided into 3 groups based on the mode of nutrition: photosynthetic protists, consumer decomposer protists, protozoan protists. Photosynthetic protists are of 3 types: chrysophytes, dinoflagellates, euglenoids. Under chrysophytes, you will have the examples of diatoms and golden algae. Dinoflagellates, that name itself, it is example. Names will be there under dinoflagellates: Noctiluca, goniolox, like that. Euglenoids, under this you have best example, euglena. Then consumer decomposer protists, the examples are slime molds. Only one example is there under that, slime molds. They have names. This is a group of organisms. So they'll have particular name of the slime molds. Under protozoan protists, you have 4 types based on the locomotory structures, which helps in their locomotion. So amoeboids, flagellates, ciliates, and sporozoans. We'll see all of them in detail. I hope all of you understood this classification. Yes, any doubts so far? Did you take it down? Yes. Sanjana, Anunaya, yes. Aditya, no. Ha, okay. Yes. Okay, fine. So once you know this classification, now let us proceed with the first one, the group of organisms chrysophytes. The first ones. Under that you are going to learn the diatoms. These are the diatoms. The first group of organisms, chrysophytes. The best example, diatoms. Under that, I have taken the picture of diatom like this. They will be looking like a soap box. They will have a soap box kind of things. Upper plate and lower plate will be there, or upper shelf and lower shelf. Two halves will be there. The two halves will be closed like a soap box. You know, lower plate or lower half or shelf will be smaller, upper half will be bigger because it should act like a lid. So it is made up of two halves, okay, that halves will be together like a soap box. So it looks like a soap box basically, and this is nothing but the cell wall. The cell wall is made up of mainly silica. So cell wall of diatoms, it is made up of silica. Not only silica, other materials will be there, but main material is silica. So siliceous cell walls are present in the diatoms. And if you open in the center somewhere here, nucleus will be there. If you open it, nucleus and other cell organelles, membrane bound cell organelles, all of them will be present in this diatoms. Remaining all structures is same. And as they are performing photosynthesis, they have particular pigments also. The particular pigments which are present in these are chlorophylls, okay, xanthophylls. Pigments are present like xanthophylls, chlorophylls. Carotenoids. You will be learning which pigments, everything you will be learning after coming here. So chlorophylls and carotenoids, they are the pigments which are present in them to perform photosynthesis, and they will have different kinds of reserve food materials. Okay, and these diatoms, they are present in both fresh water as well as marine water. They are present in both fresh water and marine waters. And these are called as chief producers in the ocean because maximum amount of photosynthesis is done by these ones. Chief producers of oceans or in oceans are the diatoms because maximum photosynthesis, maximum food preparation for the earth is done by the diatoms. And once the diatom is dead, after the death, this siliceous cell wall, silica containing cell walls will sink to the bottom of the water bodies. Once they sink to the bottom of the water bodies, these silica containing cell walls, they will form the diatomaceous earth, which is also called as Kieselguhr. This is used for various purposes, for polishing, for filtration of the syrups. So it is used as an abrasive material. So that diatomaceous earth is formed when the diatoms are dead and when this silica containing cell walls are sinking to the bottom of the pond or water bodies, they will form the diatomaceous earth, which is also called Kieselguhr. It has so many uses. So we are collecting the diatomaceous earth and we are using it for different purposes. And the maximum photosynthesis is done by these, so they are called as chief producers of oceans. And the body will be looking like a soap box. It will have upper half and lower half. They have the names and all, but it is a normal protist which can perform the photosynthesis. It will take the light with the help of pigments. Pigments will be there, different pigments, and that pigments will be taking light energy and they will perform photosynthesis. They will prepare food. And that food for future use, they will be storing it in the form of reserve food materials also. But the best part about diatoms, they are the chief producers in the ocean, and from them we are getting diatomaceous earth because their cell wall is made up of silica and other materials also will be there. Have all understood? Yes, ma? Yeah, any doubts you have in this? Diatom, it looks like a soap box. Okay, no doubts. Okay, fine. Understood, very good. So let us proceed to the next group. After diatoms, we have the dinoflagellates. Dinoflagellates. Let us see that. You can see here, this is the dinoflagellate. This also has two halves, but these two are unequal halves. Okay, they are also unequal halves, but you can't find that much difference there. But here you can find the difference. Unequal halves will be there. And these two halves will be connected in the center. Like this, one groove will be there. Upper half and lower half. Upper half is generally small, lower half is big. And there is a groove in between the upper half and lower half. That is called as girdle or cingulum. The transverse area, horizontal one, it is called as girdle or cingulum. That is the transverse groove. And here in this groove, you will have two flagella. One flagellum is longitudinal, it will be like this. One flagellum will be transverse flagellum. Two flagella are there, that is why they are called dinoflagellates. Di means two, having two flagella. So two flagella will be, one is transverse flagellum, another one is longitudinal flagellum. So as they contain two flagella, these are called as dinoflagellates, and both the flagella will be there in the transverse groove. Okay, the transverse groove will be present in this center, not exactly center, little bit towards upper side only. So in that area you will have one transverse flagellum like this, one longitudinal flagellum will be there. So they will be showing the rotation moments like this. The upper flagellum will be rotating like this, longitudinal one, transverse one will be rotating sideways. So it will be showing the whirling like movements, it will be whirling. So these are also called as whirling whips. Because of that flagellar movement. One flagellum will be rotating like this, another flagellum will be sideways rotation. So they will be showing the whirling movement. So they are called whirling whips. That is the name, okay, dinoflagellates. Now, if you see here, they have cell wall. This is nothing but what you see, the brown one is a cell wall. And this cell wall is made up of very much stiff cellulose plates. It is made up of stiff cellulose plates. They will be looking like an armor. You know, soldiers will be wearing the armor like that. So because of these stiff cellulose plates, it will be very tough or rigid on the external side, outside. So these are also called as armored dinoflagellates because they have extra protection, no? So these are also called as armored dinoflagellates. Now in this pic you can see that it is brown in color, but there are red, yellow, green, different colors are there based on the pigments present in them. Here also different pigments are there because they can perform photosynthesis. Carotenoids are present, chlorophylls are present, mainly carotenoids, because that brown color, yellow color, red color, okay, green color, blue color, different pigments present in them will give them that color. Okay, and these dinoflagellates are also producers in the ocean because they prefer mostly marine habitats. They are mostly marine. But when you take the photosynthesis of diatoms and dinoflagellates into consideration, diatoms are the primary producers. These ones are called as secondary producers after them. Second ones. The first rank goes to diatoms, second rank goes to dinoflagellates. So these are second primary producers after diatoms. Chief producers or primary producers are the diatoms. After diatoms, dinoflagellates will be coming under the primary producers category and in the oceans. They also will perform that photosynthesis and they synthesize their food material and they store that food material in the form of reserve food materials. Reserve food materials also will be different types will be there. They will be storing them. And these ones will multiply so much. Rapid multiplication will happen in the sea, oceans, marine waters. And due to that, wherever they are multiplying so much, it will cause the red tides. They'll be having the pigments, no, and their rapid multiplication will give color. That water will appear colored. So these are the ones which causes the red tides. Red tides are caused by these because of the rapid multiplication. And when they're multiplying in that water, they will be releasing different kinds of toxins, like neurotoxins will be released. Okay, so different kinds of toxins will be released by the dinoflagellates into the water bodies. If any aquatic organism or any organism, if it drinks that water, so they will be getting that toxins entering into their body, and it might kill that organisms. They are that much dangerous. Saxitoxin, digitoxin, different types of neurotoxins will be there. They will be released into the water bodies. So they are dangerous. We can't drink the water with the toxin which is released by the dinoflagellate. Even for the cattle, it is dangerous. So the dinoflagellates, though they are the primary producers, though second in number, they are good because they are synthesizing the food by photosynthesis. But we can make use of that food for other purposes. But the rapid multiplication is causing the red tides where the toxins will be released. That is dangerous for the living organisms in the water bodies. Okay. Have all understood dinoflagellates? Okay. Dino. Dino means two flagella. It's not dinosaurs. Dino. Di means two. So two flagella. Dinoflagellates. Clear? Yeah, dino means having two. Di, number. Yes, very good. Now, in the classification, the first page I have shown you diatoms, then dinoflagellates. Yes. Goniologs, yes, they are the examples which are released in the toxins. Okay, that is the example name, goniologs. Yeah, gymnodinium, all those things. After coming here, you will be learning. That much is not required for you right now. Okay, right. So after dinoflagellates, you have the third group of photosynthetic protists. Okay, bioluminescence, yes, that's correct. Avinash, very good. After coming here, you will be learning all those. Okay. The next one, euglenoids. If you want, yeah, you can write that. Examples here. Goniologs, gymnodinium. Gymnodinium. These are the examples. Okay, one more, Noctileuca. He told that bioluminescence showing dinoflagellate is Noctileuca. Bioluminescence, it emits light. Okay, that is bioluminescence. For that example is Noctileuca. You will be learning after coming here other details. This much is enough. So diatoms are over, dinoflagellates are over. The third category, okay, euglenoids. Okay, let us see the euglenoids. Under that you have only one example, that is euglena. Euglena, this is the euglena one, which is having the flagella. You can see here two flagella will be coming, but one flagellum is stopped here only and one flagellum only will be coming out. So it will be having one short flagellum and one longer flagellum, but the shorter flagellum will be stopped here only and longer flagellum will be coming out with the filament outside. Then this organism will be having the nucleus. This is the nucleus and nucleolus is there, and to the nucleus you can see mitochondria. All eukaryotic features are there. ER, Golgi, okay, contractile vacuole here near to this reservoir. This part is called as reservoir. Okay, this is called as cytopharynx. It is equal to mouth, pharynx, and this is the reservoir. So in the reservoir next into the cytoplasm, contractile vacuole is present, surrounded by the axillary vacuoles. And this is Golgi, this is ER, and these are the chloroplasts. These are the main ones. And you can see here stigma or eye spot will be there next to this photoreceptor. This is the stigma or eye spot, paraflagellar body. These are the parts of euglena. Okay, the main part with this chloroplast, it can perform photosynthesis. Photosynthesis. And this contractile vacuole is meant for osmoregulation. This stigma or eye spot or photoreceptor, these are going to receive the sunlight. They are going to identify the light source and they will be moving towards the light. So if light is available, the euglena will be performing photosynthesis and it will be acting like a plant. If light is not available, then it will act like a heterotroph animal and it will be ingesting the animals through this, which will be taken inside. So it can show both the modes of nutrition, like plant or like the animal. So it is showing the mixotrophic mode of nutrition. If light is available, then it will be showing the photosynthesis. If light is not there, it will be showing the heterotrophism. So based on that light source, it can be a phototroph or a heterotroph. So this will show mixotrophic nutrition. Mixotrophic nutrition. Okay, when light source is there, that light will be recognized by the stigma eye spot photoreceptor and it will move towards the light source to perform photosynthesis. If it is not there, it will be ingesting holozoic mode of nutrition. It will take away the smaller ones, protozoans and all inside through this cytopharynx, reservoir, okay, cytostome like that, and it will be finally sent to the cytoplasm. Here it will be forming a food vacuole. And in the food vacuole, the digestion of that whatever the protozoan food material is there, it will be done and the digested materials will be absorbed into the cytoplasm because cytoplasmic streaming will be going on. And this ER and nucleus will be there and contractile vacuole will do the function of osmoregulation because these are generally present in stagnant fresh waters. Stagnant water also they can be there, fresh waters also they can be there. But they will never be present in the sea water, ocean water, marine waters. They generally stay in the fresh waters like ponds, lakes, etc. Whereas dinoflagellates, they are mostly marine. Diatoms, they are present in fresh water as well as sea water, both. Dinoflagellates mostly marine. Euglenoids, they are freshwater organisms. These are similar to plants, because they contain the same pigment like plant, that is chlorophyll A. Particularly chlorophyll A is present in euglena, which is similar to the plant pigment. In the plant also chlorophyll A pigment is there, in the euglena also chlorophyll A is there. So in terms of pigments, euglena and plants are same and they both will perform the photosynthesis same way, okay, with the help of light. Photoautotrophism will be shown. But this is called as biological puzzle because if light is there, it is behaving like a plant. If light is not there, it is behaving like an animal. So this is actually called as biological puzzle. Biological puzzle. It was a wonder for the scientists first, how it is able to do. Biological puzzle. Okay. This is about euglenoids. Euglena is the best example, euglena. I hope all of you understood. So we have completed the photosynthetic protists. We have completed the photosynthetic protists. Yes. First one diatoms, second one's dinoflagellates, third one's euglenoids. Okay, they have different pigments, but all of them will perform photosynthesis and they will be preparing the food. Yes. Very good, very good. One last point. In diatoms, you know, the cell wall is made up of silica. In dinoflagellates, cellulosic plates are there. Stiff cellulose plates are there. In euglenoids, there is no cell wall. No cell wall. This external layer is called as pellicle. It is made up of protein rich layer. Cell wall is absent in euglenoids. They contain a protein rich layer called as pellicle. Pellicle is present in them. Okay. Army by goniologs. Goniologs is the dinoflagellate. Okay. So diatoms, they are made up of silica in their cell walls. Dinoflagellates, they have cellulose in their cell walls, cellulose plates. Euglena, no cell wall, but pellicle is present, which is a protein rich layer. It is made up of proteins. Okay. What is that? Golden algae. Yeah, golden algae, their name is desmides. That is also, they also look like diatoms only. In your textbook, they have given only diatoms. Golden algae, example name is enough. Okay. Yeah. Yeah, any further doubts, ma? Have all understood? Silica contains carbohydrates. Silica is that crystals will be there in the sand and all. Silica is a chemical. Yeah, yes, Tikshana. Anju. Okay. Yeah. Okay. Shall I ask questions now? No doubts. Very good. Slime moles. Yeah, we'll proceed to that. We'll proceed. Okay. Time is, time is not there. So I'll ask the questions tomorrow. I hope all of you understood photosynthetic protease. Okay. Let us go to the next one. I'll ask at the end. Slime moles. This is how the slime mole's body will be looking. This is slime mold. Okay. These are the ones which are decomposer protists, particularly decomposer and consumer protists. So they are particularly the saprophytes, saprophytic protists, because they'll be feeding on the dead and decaying organic matter. So whatever organic matter that is present on their way, they'll be eating them. Twigs, organic matter, they'll be feeding on them and they'll be moving. Okay. And during the favorable conditions, they'll form this structure which is called as plasmodium. This structure is called as plasmodium. This is the body of the slime molds. It is the body of slime molds. So body of slime molds is called as plasmodium. So during favorable conditions, when it is feeding on the twigs, the fallen leaves, organic matter, this plasmodium will be formed and it will be growing to several feet. It can become so much big, okay, by consuming all the organic matter, whatever comes on its way. So this slime mold body, plasmodium, okay, it will be growing in the favorable conditions, and during unfavorable conditions, it forms the fruiting bodies. They form fruiting bodies. And in that fruiting bodies, spores are formed. And spores will be dispersed by air. Air currents will be dispersing the spores. Okay. And these are all nuclei. You can see here, these are all nuclei, multinucleate condition. So plasmodium is having multinucleate condition. Many nuclei will be there, multinucleate condition. So this slime molds, just they will be looking like this. The body is called as plasmodium and it will be spreading by feeding on the organic matter and twigs. So it is saprophytic in nature, and during unfavorable conditions, this plasmodium will give rise to the fruiting bodies like this, and in that spores will be formed like this. This is the fruiting body and the spores will come out. And the spore will germinate during the favorable conditions. This is the slime molds. The body of the slime mold is Plasmodium. I hope all of you understood. Okay. The last one, protozoans. Protozoans. These may be there in the fresh water or marine waters. They can be predators or parasites, and they show generally heterotrophic mode of nutrition. There is no autotroph in this, all are heterotroph. And in this, the first example, amoeboid protozoan, in the classification I have given you, amoeboid protozoans. Amoeba, it has the locomotory structures that are pseudopodia. Pseudopodia. So they don't have any true legs like that, but false feet are there, pseudopodia. These are called false feet. Okay. So they'll move from one place to another place with the help of pseudopodia. Amoeba, you know, irregular in shape, and it has one contractile vacuole, okay, meant for osmoregulation. Then you have some of the amoeboids may have silica in their cell walls. Marine forms will have that. Again, one more example is there, entamoeba. Amoeba, entamoeba. These all will come under amoeboids. And after this, you have the paramecium. Paramecium will have cilia. The locomotory structures in this are cilia. So it is coming under ciliated protozoans, because they will be moving from one area to another area with the help of cilia. So ciliated protozoans. These are also there in the fresh waters generally. Then this is also called as slipper animalcule because of its shape. Okay, this also has the contractile vacuoles. See the number of contractile vacuoles here. Posterior contractile vacuole this one, and anterior contractile vacuole is this one. Two contractile vacuoles are present. Then this is the structure of Plasmodium. This is a sporozoan. In this, there are no locomotory structures. No locomotory structures, because they don't move with any locomotory structures. Okay, they don't have, so they won't move. And this Plasmodium is the causative agent of malaria. It is a pathogen which causes the malaria, so it is called as Malarian parasite. Malarian parasite. The structure of Plasmodium will be looking like this. Body of the slime molds is the Plasmodium, that is the name of the body. But here this is the Malarian parasite. Malarian parasite. Okay, it is causing the malaria, and it does not have any locomotory structures. Then you have the last one, the flagellated protozoans, which are having flagella for their locomotion. Flagella they contain for locomotion, and example for this is trypanosoma. Trypanosoma. Amoeboid protozoans, amoeba is example, entamoeba. Then ciliated protozoans, paramecium, because they move with the help of cilia. Then flagellated protozoans, example is trypanosoma. Then there are no locomotory structures in the sporozoans. For that sporozoan, example is Plasmodium. Okay, and it is the Malarian parasite. I hope all of you understood. Yeah, trypanosoma, the name of the organism, flagellate. It causes sleeping sickness, trypanosoma, African sleeping sickness. Yeah, have all understood? So all of you, as time is not there, I would like to end here. Tomorrow I'll ask questions based on this. read that Protesta and come tomorrow. Any doubts? Yeah, sleeping sickness is caused by trypanosoma. No doubts. No doubts, all of you? Give a thumbs up. Understood everything? Yes. Okay. Doubta. Okay, what is that? Yes. Okay. Tomorrow I will clarify the doubts if you have further. Okay. So all of you read and come Protesta. Tomorrow we'll proceed to the next topic. Thank you, ma. See you tomorrow.
Hello, hello, hello everyone. How are you all? Good morning, all of you. Good morning, all of you. How are you all? Good morning, Navyashri. Madhu, Mita. Good morning, beta. Deeptika, Tikshana, Lakshmi, Ratna, Lekhya, Neer. Everyone, a very good morning to all of you. Thank you. Thank you so much. So I am good, beta. How are you all? Mr. Adnan, I'm good, beta. Here comes our favorite teacher, Madhuri. Thank you, Ratna, Lakshmi, Sanjana, Satyam, Parheen, Sujata, Pujita, Shravan. Okay, everyone, a very good morning to all of you. I hope you all are doing great, and let us start today's class without any further delay. Okay, in today's class, we are going to talk about the chapter breathing and exchange of gases. So yes, everyone, I think this chapter has been done. Just like I have done the animal tissues, I have discussed animal tissues previously with you. Just like that, I think breathing and exchange of gases is also, taught a little bit. Yes, yes, everyone. Malika Arjun, Shravani. Yes, thank you. Yes, all of you are excited. So yes, tell me quickly, what have you already understood in this chapter, breathing and exchange of gases? Yes, in the comments, tell me quickly. Yes, tell me quickly in the comments. Don't talk to each other, talk to me, okay? Don't talk to each other. So we are going to understand about the chapter breathing and exchange of gases. Breathing and exchange of gases. This is the name of the chapter, breathing and exchange of gases. Yes, what you have studied already in this chapter. Okay, you have studied inspiration, expiration. Okay, all of you, you have studied inspiration and expiration. Yes. You have also studied alveoli exchange of gases. Okay. Okay, so we will see what we can discuss today from this chapter. Okay. We will see what we can discuss. So first of all, I will give you a brief introduction about the chapter. What all things are there which you have to deal with? What all things are there which you have to study in this chapter? Let me tell you first of all that. Okay, everyone? Okay, everyone. Sindhu, it's okay, beta. So now all of you are into the class. Just focus on the class, beta. Okay, don't waste your time in chatting with each other. Just stay focused and listen to the class, because each and every minute of yours is important. Okay, okay. Yes, everyone. Okay. So in this chapter, beta, first of all, let me tell you what is the meaning of the name of the chapter. Okay. So there is a process in our body. That process is known as respiration. Yes. What is that important process? That important process is respiration. Okay. By this process only, by the process of respiration only, we are getting energy. So beta, we are getting energy. Okay. See, two things we require for the respiration. We require food and we require air. Yes or no? Air and food are very, very important na, for our survival. We cannot live without food. We cannot live without air and also water. Yes or no? Water is also required. But if we see only food, then from the food mainly we are getting glucose. Okay. What is glucose? Glucose is C6H12O6. And from the air, this we are getting from the food. And from the air we are getting oxygen. Okay. So in presence of oxygen, this glucose will break, beta. Breakdown of glucose in presence of oxygen. This is cellular respiration. What it will yield? What it will form? When this glucose will break down in presence of oxygen, this will lead to the formation of carbon dioxide, water, and lots of energy. Energy. For this energy only, this respiration is taking place. Okay, for this energy, you will study the cellular respiration in botany in detail. Okay, cellular respiration. Here I will talk about this whole process. This whole process is occurring in 5 steps, beta. This respiration, this whole process is a 5-step process. It is a 5-step process. Respiration is a 5-step process, beta. So yes, everyone, can you tell me the first step? Can you tell me the first step of this process? Can you tell me the first step of this process? Yes, everyone. Tell me in the comments. Tell me, be active in the chat section. Tell me in the comments, the first process is pulmonary ventilation. First process is breathing. Yes, absolutely correct. The first process is beta breathing. This breathing is also known as beta pulmonary ventilation. Pulmonary ventilation. So in this breathing or in this pulmonary ventilation, two processes are occurring. One is inspiration, another is expiration. Yes, inspiration can also be called as inhalation. Inspiration can also be called as inhalation and expiration. There is a separate mechanism how inspiration takes place and how expiration takes place. I think you have studied the mechanism of inspiration and expiration previously. Yes or no? Have you studied the mechanism of inspiration and expiration previously? So in inspiration, we will take the air inside, and in expiration, we will throw the air outside. When beta we are taking the air inside, in this air, there is a very important gas, that is oxygen, right? So we want to take that oxygen, okay? So how that oxygen from the alveoli go into the blood And how from the blood the CO2 will come out in the alveoli? That is known as exchange of gases. So what is the second step? Everyone tell me in the chats quickly. What is the second step? Second step. What is the second step? Exchange of gases. At which level? This exchange of gases is occurring at which level, beta? In between what and what? Exchange of gases between alveoli and blood, because in the alveoli is the part of the lungs. You have a respiratory passage. For the respiration, you require respiratory passage. Have you studied about the respiratory passage, everyone? This respiration, this whole process is carried out by the respiratory passage. There is a respiratory passage. If you know respiratory passage, I will tell you briefly. In this respiratory passage, it starts from your external nostrils. We have above the lips, above the upper lip, we have these two nostrils. Okay, these are external nostrils or known as external nares. From here, our respiratory passage will start. It will go to the nasal cavity, then it will go to the nasopharynx. Okay, then larynx will come here, voice box, larynx, from which I am speaking. Yes, I am audible to you all. Okay, I am producing sound. So that is sound or voice box. Okay, so then that larynx will then further lead to the trachea, and that trachea will divide into two parts and it will enter into the lungs. So we have two lungs. Okay, we have two lungs. So the trachea will divide and it will form bronchi. Further, it will form the secondary bronchi, tertiary bronchi, bronchioles. Okay, so trachea, primary bronchi, secondary bronchi, tertiary bronchi, then bronchioles, and then alveoli will come into the lungs. Whole this passage is respiratory passage. In this respiratory passage, two parts are there, beta. Two parts. Two parts are there. One is the conducting part. One is the conducting part, and another is the respiratory or exchange part. Another is the respiratory or exchange part. So this respiratory or exchange part is alveoli, beta, alveoli. Before that, everything is the conducting part. Okay, so conducting part starts from external nostrils. It starts from external nostrils till terminal bronchioles. Whereas in the exchange part, there are respiratory bronchioles. Respiratory bronchioles will come after terminal bronchioles. Respiratory bronchioles till alveoli. Till alveoli, beta. Till alveoli. Okay. Till alveoli. So this is our respiratory passage. Respiratory passage. I have briefly explained you the Yes, everyone. Is there any lagging still? Is there any lagging still, everyone? Everyone, please tell me, is there any lagging still? Or am I audible, visible to all of you? Now it's clear? Okay, everyone, just coming to the class. Yes, everyone. Today MSET exam. Oh, I didn't knew. Okay. All the best to those who are giving MSET exam. Okay, I know you people will not be giving MSET exam, right? So just focus on the class. Everyone focus on the class. Yes, everyone. Now it's fine. It's clear. It's okay. Everything is okay. Now just come back to the class and focus. Okay, what I was telling you that the process respiration occurs in the 5 steps. First step is breathing. Breathing simply means inspiration and expiration. Inspiration and expiration. I was telling you the second step, that is exchange of gases. So first of all, the exchange of gases will occur in the alveoli beta, inside the lungs alveoli. And do you know, around the alveoli so much of blood vessels are there. Okay, so in those blood vessels, what is there? What is flowing? Blood is flowing. Okay, so this exchange of gases will occur between alveoli and blood. Okay, alveoli and blood. We can say at alveolar level, the exchange of gases is taking place. Now, in the blood, oxygen has come into the blood. Okay, now this oxygen needs to reach to every tissue. So the third step will be, yes everyone, can you tell me in the comment? Can you tell me in the chat section what will be the third step? The oxygen should go to the tissues. So we will call transport of gases. Yes everyone, transport of gases. Transport of gases. In this transport of gases, you know that oxygen is going from alveoli, it is going to the blood, and from blood it is going to the tissues beta. It is going to the tissues. This is oxygen. Oxygen is going from alveoli to blood, from blood to tissues. But if you see carbon dioxide is coming from tissues to blood and then from blood to alveoli. So this is transport of gases beta. Transport of gases. Alright, are you understanding the meaning of transport of gases? And the medium of transport is beta blood. The medium of transport is blood. The medium of transport is blood. Blood is the medium of transport. Through the blood only the oxygen will travel. Through the blood only the carbon dioxide will travel. Both of the gases will travel through the blood. I will tell you in which forms they will travel. Okay. Okay. Now so now tissues got the oxygen. Tissues are very happy. Tissues have got the oxygen. Okay. So here again, when tissues will get the oxygen, from the blood to tissues exchange of gases will occur na. So what can be the fourth step? Everyone, fourth step. Fourth step is again exchange of gases. Again exchange of gases between blood and tissues. Between blood and tissues. Okay, between blood and tissues. And now the tissues will get the oxygen. So what will be the fifth step? Fifth step will be cellular respiration. Cellular respiration means utilization of oxygen for the breakdown of glucose. For the breakdown of glucose to get energy. So are you people understanding the 5 steps, everyone? If you are not understanding any of the step, please tell me in the comments. Yes, everyone. So the first step is breathing, also known as pulmonary ventilation. Okay, also known as pulmonary ventilation. Inspiration, expiration. Second step, when oxygen will go into the alveoli, that oxygen needs to be exchanged and it needs to go into the blood. So now oxygen has reached into the blood. Then this blood will transport this oxygen to the tissues. So when this blood will reach to the tissues, again exchange of gases will occur between the blood and tissues. And then the tissues will get the oxygen and then the tissues will utilize the oxygen. This is going to be the 5 step. Yes, Ratna, third and fourth, what is the difference between third and fourth? Beta, transport. See, everyone, exchange means, I will tell you. Wait, let me go to the next. See, this is alveoli beta. And let us suppose this is blood. This is blood. Okay, this is your blood and this is alveoli. This is alveoli. So, exchange means the oxygen and carbon dioxide is getting exchanged on the basis of the partial pressure. Okay, if oxygen partial pressure is more in alveoli and less in blood, oxygen will come into the blood. Okay, see here in alveoli, partial pressure of oxygen is beta 104. This blood which is coming here na, this is beta deoxygenated blood. In this blood, oxygen is not there. So this deoxygenated blood partial pressure of oxygen is 40. Partial pressure of oxygen is 40. So oxygen is going to come from alveoli to blood. Oxygen is coming. This is diffusion or exchange of gases. Oxygen is coming. Now if you see carbon dioxide, this is deoxygenated blood beta. Carbon dioxide is more. Partial pressure of carbon dioxide in this blood is more. It is 45. Whereas in alveoli, partial pressure of carbon dioxide is 40. Okay. So, where it is more, here it is more. And where it is less, here it is less. So, that is why carbon dioxide is going from blood to alveoli. Beta, this is exchange. This is exchange. And this exchange of gases is occurring at alveolar level. Are you understanding, everyone? Are you understanding this? Exchange at alveolar level. Now I can tell you exchange at tissue level. Fourth step. Fourth step. Okay? Fourth step. So now let me tell you the fourth step. Let us suppose this is the blood. This is beta blood. Which blood is coming near to the tissues? Near to the tissues, it is oxygenated blood. And let us suppose these are the tissues. Beta, these are tissues. Okay. In the tissues, carbon dioxide will be more. Partial pressure of CO2 is more beta, 45. Okay. And partial pressure of oxygen is less. Partial pressure of oxygen is less. Okay. In the oxygenated blood, partial pressure of oxygen is more, 95. Whereas partial pressure of carbon dioxide, is less, 40. Okay. And what is the partial pressure of oxygen in the tissues? 40. So, you can see, first of all, oxygen. Oxygen is more in the blood. Oxygen is more in the blood. Oxygenated blood, 95 mm of Hg oxygen. In the tissues, how much oxygen? 40. Where it is more? Blood. Where it is less? Tissues. So, oxygen will exchange from blood to tissues. Oxygen will come. Are you understanding? Next. CO2. Where CO2 is more? Tissues. Where CO2 is less? Oxygenated blood. So, CO2 will diffuse. CO2 will diffuse from tissues to blood. CO2. This is exchange. Ratna, are you understanding the exchange? Now I will tell you the transport. In which form the oxygen and carbon dioxide will be transported? Oxygen is transported from here to here and CO2 is transported from here to here. Okay. So that will be transport beta. Exchange is simply between the two things on the basis of difference in partial pressure. Okay. On the basis of difference in partial pressure. All these partial pressures you have to remember. In the NCERT there is a very nice figure in which all the partial pressures are mentioned. And it is very easy to remember the partial pressure. Once you will come here, I will tell you the trick. Very easy trick to remember the partial pressure. Okay. So now everyone, you understood the exchange na? Here this is the exchange between alveoli and blood. Here is the exchange between blood and tissues. Now let me tell you the transport. Let me clear you the meaning of transport of gases. Okay. Okay. Come here. Transport of gases beta. Transport of gases. So we have two gases, one is oxygen, one is carbon dioxide. Both the gases will be transported by blood. Blood is the only medium in the body for the transport. Ammu, beta, what is diffusion? Beta, diffusion is also exchange of gases. Exchange of gases is occurring by simple diffusion. Diffusion means whenever something is going along the concentration gradient. When something is going from high concentration to low concentration. Okay. So when a gas is moving from high partial pressure to low partial pressure, it is diffusion, beta. It is diffusion. Now everyone, coming to transport of gases. What is the medium of transport, beta? Blood. Blood. Okay. I have told you, I remember I taught you blood. Yes, everyone. Do you remember blood? Do you remember blood? Yes, everyone. Tell me, do you remember blood? What are the components of blood? Blood has plasma. Blood has formed elements. Formed elements are RBC, WBC, platelets. Do you remember? Yes, everyone. So, in which cells there is hemoglobin? RBC. In which cells there is hemoglobin? RBC. So, RBC and plasma, these two will help. The plasma will help and the RBC will help in the transport of gases. In this transport of gases, WBCs will not help, platelets will not help. Okay, WBCs will not help, platelets will not help. Only two things will help. One is plasma and another is RBC. Okay, everyone. Now, blood. Let me tell you the transport of oxygen and transport of carbon dioxide. Both I will tell you. Transport of carbon dioxide first. Transport of carbon dioxide occurs in three forms, beta. Three forms. Whereas transport of oxygen occurs in two forms. In two forms. Okay, what are these two forms and what are these three forms? What are these three forms? First is this CO2 and this oxygen, gases can get dissolved. And what is the fluid part of the blood? Plasma. So these oxygen and carbon dioxide can get dissolved in the plasma. Okay, so the first form is dissolved form, beta. Dissolved form. Dissolved form. How much percentage of CO2 travels in the dissolved form? 7%. 7%. This dissolved form travels through plasma, beta. 7% CO2 travels, 7% CO2 travels in the dissolved form and this travels through plasma. Through plasma. Now, next is bicarbonate ion form. Bicarbonate ion form. What is a bicarbonate ion? Bicarbonate ion is HCO3 negative. In this ion form, 70% of the CO2 travels in this form, beta. 70% of the CO2 travels in the form of bicarbonate ion. Now, can you tell me ions are present in which part of blood? Plasma. Yes, ions are present in which part of blood? Plasma. So, this bicarbonate ion form also travels through plasma. It travels through plasma, beta. Through plasma. Through plasma. So, total, how much percentage travels through plasma? Total, how much percentage travels through plasma? Yes, tell me. Total, how much percentage travels through plasma? can we tell 77% CO2 travels through plasma? Travels through plasma. Yes or no, everyone? How much is left? 23% is left. So 23% is left. Okay. That will go into the RBC in form of carb-aminohemoglobin. Carb-aminohemoglobin form. Carb-aminohemoglobin form. How much percentage left? 23%. Okay, 23%. HbCO2 you can write. HbCO2 is carb-aminohemoglobin form. How much percentage? 23%. Okay, hemoglobin is present inside the RBC. So it will not go through the plasma. It will go through the RBCs. 23% through RBC. Through RBC. So everyone, CO2 is getting transported by blood from one place to another place. Transport simply means from one place to another place. When you will be coming from your hometown to this campus, Sri Chaitanya Goshala, how you will be coming? Some people will be coming by public transport, some people will be coming by bus, some people will be coming by train, some people will be coming by their own vehicle like their car. Okay, some people will be, maybe some of you are nearby only, so they will come by bike or scooty. Everyone has different mode of transport, na beta? But in our blood, there is only one mode of transport, that is blood. And in blood, only two things are there. One is plasma and one is RBC. One is plasma and one is RBC. So, through plasma, how much CO2 will travel? 77%. 7% in dissolved form, simply it is dissolved. 70% in bicarbonate ion form. I will teach you when I will get further class. So I will teach you that how this bicarbonate ion is getting formed in detail. Okay, but first of all you should understand this. And then how much percentage through the RBC? Inside the blood only RBC is there, na? 23% CO2 will go inside the RBC. It will tell, no, I don't like plasma. I want hemoglobin. I want to sit with hemoglobin. So, this 23% CO2 will sit with hemoglobin. Okay. And then it will form the carb-aminohemoglobin. So, are you understanding this? 3 forms. Are you understanding all these 3 forms? Everyone, please give me a thumbs up if you are understanding these 3 forms. Now, I will tell you very easy 2 forms of oxygen. Oxygen will travel only in 2 forms. Okay, only in 2 forms. One is the dissolved form, beta. One is the dissolved form. And one is the oxyhemoglobin form. Oxyhemoglobin form. Oxyhemoglobin form. Dissolved form, oxyhemoglobin form. Oxyhemoglobin form. How much percentage? Dissolved form 3%. 3% oxygen in dissolved form. Rest 97% oxygen with hemoglobin. It will tell, no. I to like only hemoglobin. Okay. I will not convert into any ion. I will like hemoglobin. I will go with hemoglobin. So 97% oxygen goes through the RBC with the hemoglobin. So, this is through RBC. This is through RBC and this is through plasma. This is through plasma. Dissolved in the plasma. Dissolved in the plasma. So, are you understanding? Yes, everyone. Are you understanding this? Are you understanding the 3 forms of CO2 and 2 forms of O2? Yes, everyone. So, now you understood the meaning of breathing, inspiration, expiration. exchange of gases between alveoli and blood, transport of gases, transport of oxygen, transport of carbon dioxide, in how many forms, what is the percentage, and exchange of gases between blood and tissues, and then cellular respiration. Ultimately, the last step is cellular respiration. Yes, everyone, tell me, have you understood the glimpse of the chapter? Have you understood what all we are going to study in this chapter? Obviously, beta, I cannot teach you the complete chapter in detail in 45 minutes, hai na? I hope you are understanding this, but at least I am trying to make things easier for you. When you will come to the campus, when we will teach you this chapter, it will take almost 1 week. Okay? At least 4-5 classes are required. Okay, but when you will listen to those classes now, you will understand them very easily. Why? Because now I have made easy. I have made this chapter easy for you. Now you have understood the meaning of breathing, meaning of respiration, meaning of inspiration, expiration, meaning of exchange. What is the difference between exchange at alveolar site and what is the difference between exchange at tissue site? And now you understood what is the meaning of transport. Okay. So I hope you understood all these 5 steps, the meaning of all these 5 steps. This is the essence of the chapter. This is the overview of the chapter. If you have understood these terms, the meaning of these terms, okay, then you will understand the topic very easily. Under the topic breathing, you will understand the mechanism, you will understand the respiratory volumes, capacity, how much air we are taking in, how much air we are throwing out, what is the capacity, how much total we can take in, total we can throw out, all these things you will understand. Then you will understand exchange at 2 levels, depends upon the partial pressure, depends upon the solubility, and depends upon the thickness of diffusion membrane. I have told you diffusion membrane, yes, in the animal tissues. Do you remember diffusion membrane in the animal tissues? Yes, everyone, tell me. Okay, now quickly I will ask you one question. Quickly I will ask you one question. I have told you here transport of gases, okay? So can you tell me total how much percentage of CO2 is getting transported by RBC? Yes, without looking at the board you have to tell. So without looking at the board you have to tell. How much percentage of CO2 traveling through RBC? 23%. Okay. How much oxygen? How much oxygen traveling through RBC? How much total oxygen traveling through RBC? 97%. Okay. Now I will ask you how much oxygen traveling in dissolved form? In dissolved form. How much oxygen in dissolved form? 3%. How much oxygen in dissolved form? 3%. How much CO2 in dissolved form? How much CO2 in dissolved form? How much CO2 in dissolved form? 7%. Why? 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 This is the question. Why 7%? Why 7% CO2 in dissolved form? And why 3% O2 in dissolved form? Why? Why? Why more CO2 is getting dissolved in the plasma than oxygen? Sampath, I know, because I want to, boost up the confidence of my students. Why? Tell me why. You told me 7% CO2 and 3% O2 in dissolved form. Why? That is my question. So I will tell you the answer because CO2 is 20 to 25 times more soluble than oxygen. Solubility of CO2 is more. This is because of the solubility. Solubility of CO2 is more than the solubility of oxygen. Okay, solubility of CO2 is more than the solubility of oxygen. That is why CO2 is going 7% in dissolved form and O2 is going only 3% in dissolved form. Are you understanding? Okay, so that was my time and that was all about my class, and I hope you all have understood all these basic things and I hope you have enjoyed my class. Okay, so just don't forget to hit the like button. Okay, subscribe to the channel, hit the like button, like all the videos, okay, which you are seeing and getting the benefit now. You should like those videos. Okay, that is the all motivation you can give to us, and thank you so much for, okay then, bye bye, take care, God bless you all.